Uh, this is Cynthia. Sorry, I'm having some technical issues this morning, but thank you for attending MOT Transplant Grand Rounds. We're really excited to have Anna, if you don't mind introducing your speaker.
Absolutely, I don't mind. It is my pleasure to introduce Dr. Sergi Clotet-Freixas, who has been an outstanding postdoctoral fellow in my lab since 2016. And Sergi has been working on many projects, including antibody-mediated rejection. And in fact, his invited editorial on the topic of extracellular matrix remodeling in antibody-mediated rejection has just been accepted in top-end renal journal, JASN, yesterday. However, today, Sergi will tell you about a topic that is very close to him and that he's really pioneered in my lab. He has pushed me into the field of sex differences and really has driven all the studies that he will tell you about today. Sergi has received numerous awards, including the STARS International Award given by the American Society of Nephrology, and the Crescent Postdoctoral Fellowship, which is given by Kidney Foundation of Canada and CIHR, and which is arguably the most coveted and competitive award in kidney research in Canada. So without further ado, Sergi, please go ahead and present your talk.
Thanks, Dr. Konvalinka, for this kind introduction, and good morning, everyone. It is a pleasure for me to be here today and talk about my favorite research topic, which is based on the idea that a better understanding of the sex differences on the biology of our cells will make us better on understanding and treating human diseases. I have nothing to disclose, and as I hope to show you today, the kidney is the perfect example on how physiological sex differences may impact disease, especially when metabolism is involved in the equation. As mammals, we show a clear sexual dimorphism, which is the condition where the two sexes of the same species exhibit different characteristics beyond the differences in their sexual organs. These features include size, weight, color, behavior, and cognition, among other traits. From an evolution perspective, sexual dimorphism is partially due to natural selection, but most of it is ascribed to sexual selection. That means that in many species, males were pressured to develop a more explosive phenotype in order to compete with other males for the attention of females. Early in my career, I was fascinated to learn that the kidney shows a marked sex dimorphism, being bigger and having a higher filtration capacity in males. And even more fascinating was to see that many mechanisms are activated differently in males and females. However, the kidney is not the only sexual dimorphic organ. If we think about it, and if we think about sex and metabolism, we probably think about several physiological features that are different between men and women, such as organ size, body composition, or exercise performance. On top of that, many human diseases present a sex-based epidemiology, including diseases with an important metabolic component, such as diabetes and obesity, to liver and heart diseases, and even lung cancer. One of my goals today is to convince you that sex may have an impact on how the cells of all our different organs work, and that metabolism could be the common denominator to understand sex differences in health and disease. But let's start on how I got here. This week is special to me because exactly 10 years and 2 days ago, I left my hometown in Catalonia to embark a PhD in nephrology research in this beautiful building next to Hospital del Mar of Barcelona. As most of us, I started my scientific career facing a problem, which in this case is called chronic kidney disease. Chronic kidney disease is actually a major public health problem as it affects around 10% of the adult population, is associated with increased morbidity and mortality, and represents an economic burden. Importantly, diabetes is recognized as the leading cause of chronic kidney disease. Diabetic kidney disease is indeed responsible for about half of the cases of kidney failure. In the kidney field, growing evidence supports that male sex predisposes to a faster and more severe progression of diabetic kidney disease. However, the reasons of this sex dimorphism are still unclear. If we find these reasons, we will be able to design better therapies to prevent kidney disease in men and women. But solving the puzzle of sex differences is not easy, as many variables are involved in the equation. For example, sex differences may be ascribed to the sex chromosomes, to the sex hormones, or to both. In addition, sex hormone levels may fluctuate fluctuate and be influenced by many factors such as age and diabetes, making the study of the sex dichotomy in diabetic kidney disease even more complicated. Along these lines, the fact that female sex protection is lost, at least in part after menopause, reinforced the idea that hormones play an important role and that estrogens could exert beneficial actions over androgens. Androgens indeed seem to be deleterious, and we have seen that in the lab. In diabetic animals, different functional markers of renal injury were exacerbated by male sex and at the same time reduced by surgical castration in these diabetic males. To date, the molecular mechanisms behind the sex-based dichotomy in diabetic kidney disease remain unclear. To address this unmet need, my research goal during my PhD and now as postdoc is to elucidate the mechanisms that underpin this sex-specific progression of diabetic kidney disease. And this unmet need perfectly aligns with the current philosophy of NIH, which is trying to incentivize the inclusion of male and female individuals in our research studies. The necessity of more gender and sex balance studies is a very timely topic in the biomedical field, and this is gradually being reflected in the programs of funding institutions such as the CIHR. But before proceeding with the talk, I think that first it's necessary to stop for a second and define what is gender and what is sex, as these are two terms that we usually use interchangeably and have different meanings. While gender refers to socially constructed roles, behaviors, and identities, sex is a set of biological attributes in humans and animals. Because of my scientific background and the lines of investigations that I follow in the lab, my research is focused on the biological differences between sexes, and therefore I will employ the term sex through the entire presentation. To study sex differences, animal models are a very valuable tool. But what we find in the biomedical field is that the use of only male animal models has dominated the literature. There are many reasons for this imbalance, starting by the androgenic nature of human studies that led to the design of the experimental approaches, as many social components may influence females to be less studied than males. There are other reasons more at the biological level, such as the presumably confounding effects of the estrous cycle, and finally some logistics reasons linked to the study of both sexes in the lab, such as higher costs, higher sample size, or protocol variations. Considering sex as a biological variable will require a global shift in the science culture, which is gradually starting to cook. Classically, the study of sex differences was limited to reproductive and hormonal issues, but in the last 2 decades, new evidence emerged proving that sex also affects cell physiology, metabolism, pain perception, and even disease manifestation and responses to treatment. And then 2020 came and reminded us that there is also a sex debate when it comes to viruses and vaccines. Despite this cumulative evidence, it wasn't until the recent years when more scientists started to consider sex as a biological variable. Indeed, the increasing number of sex-focused studies is leading to novel observations that are challenging previous myths. A clear example is this editorial in the field of neuroscience with concepts that can be applied in other fields. The author claims that the assumption of female animals being more variable than males comes classically from the stereotypical idea in our society that women are more hormonal and emotional than men. To refute this assumption, the author relies on this meta-analysis in which the authors assessed more than 300 variables and demonstrated that the experimental variability in female rats was not higher than in male rats. This suggests that the estrous cycle does not increase variability, and therefore there's no need to quadruple the study groups and costs, and the cost of the study. And to minimize the number of animals and costs in the lab, it is recommended to always start with the first experiment with sex mixed cohorts and continue with this approach if no clear sex difference is observed in our readouts. In contrast, if we observe sex differences, we are forced to study both sexes as different experimental groups and ideally in parallel. And guess what? This concept also applies to the cells. In my PhD, I wanted to explore if there were any studies out there using male and female kidney cells that could help us understand why our male animals were having a more severe diabetic kidney disease than females. However, this concept is quite novel, and there's still a paucity of studies that consider the sex of the cell. Instead, some authors have explored the effect of sex hormones in certain cell types of the kidney. For example, they have seen that the treatment with the androgen testosterone promotes apoptosis in kidney cells. However, the mechanisms mediated these effects are still unclear. And this is what we try to identify in the lab. In this picture, you can appreciate how complex and diverse is the kidney nephron with many compartments and cell types. While filtration mainly takes place thanks to the team effort of several specialized cell types in the glomeruli, the tubular cells are the main responsible for the kidney reabsorption. In the laboratory, I specifically work with these primary human proximal tubular epithelial cells for several reasons. They are the most abundant cell type in the kidney. They play a key role in the development of fibrosis, which is considered the main hallmark of advanced kidney disease. They express both male and female sex hormone receptors. And also they are highly metabolically active, which probably makes them more susceptible to alterations induced by high glucose in a setting of diabetes. To explore in a global fashion the molecular mechanisms induced by sex hormones in the kidney cell, I contact Dr. Konvalinka to see if we could apply the proteomics methodology that she previously optimized and study what would happen globally at the protein level if we incubated the cells with the androgen dihydrotestosterone and the estrogen estradiol. Our hypothesis was that androgen stimulation would lead to maladaptive changes in kidney cells compared to estrogen stimulation. So I applied a SILAC-based proteomics approach to my research question, and after quantifying over 5,000 proteins, our main finding was that energy metabolism could be the key link to understand sex differences in kidney physiology and how they will predispose to a sex-specific progression in diabetic kidney disease. In particular, we found that proteins involved in critical metabolic processes such as glutamine metabolism, glycolysis, and the TCI cycle were upregulated by the dihydrotestosterone, that you can see here by the orange bars, but not by estradiol, represented by the green bars. These results suggested that androgens alter energy metabolism in human kidney cells, at least more than estrogens. But to evaluate if this finding is important for our research, first we need to revisit what metabolism is. Metabolism is the sum of chemical reactions that take place in living cells, providing energy for life processes and the synthesis of cellular material. These reactions can be classified as catabolic reactions or anabolic reactions. Our cells are made of molecules, which we can compare to building blocks of different types and sizes. While catabolism is the breakdown of complex building blocks to more simple pieces, anabolism is the opposite process. And while the breakdown of substances enables the cells generate energy for their functions, the anabolic reactions result in the production of biomass, which is needed when the cells need to duplicate some of their material before dividing, for example. To keep the metabolic balance of the cells, these processes need to fit each other. But sometimes these blocks cannot be available for these reactions, as they need to be used to build stable structures like organelles, or they need to be bound to proteins or DNA, or they are just excreted out of the cell. And you may be wondering, if that happens, how can the cells keep working? The answer is very simple. They need food. And to make new blocks from the food, which we know as metabolites, cells have a huge army of tiny movers and builders, which are the metabolic enzymes. Interestingly, we found that kidney cells exposed to androgens have higher levels of enzymes that make blocks from glucose and glutamine. Or in other words, enzymes that break down glucose and glutamine with the final goal of making energy in the form of ATP molecules. Under healthy conditions, kidney cells convert most of their glucose into an intermediate called pyruvate, and then further to ATP. Pyruvate levels are very important for a good metabolic balance because this metabolite is a splitting point between a path of glucose that generates a lot of energy and another fate, which is lactate production via glycolysis that generates less energy. If glucose is yielding enough energy, then the cells can use glutamine for biosynthetic reactions, and only a bit of it is converted to glutamate and invested for energy purposes. But to make energy, the enzymes need a very specialized machinery. Now, I'm going to ask you to imagine that the kidney cell is a car. To make a car work, we need fuel. And to make energy from the fuel, we need a motor so the car can move. But what happens if we are driving all the time? Well, we will consume more fuel, which will result more expensive, and eventually the chances of a breakdown will be higher. But those unwanted outcomes can be avoided with proper maintenance and repair of our car. In our cells, three important fuels are glucose, glutamine, and pyruvate. The motors that make the energy are called the mitochondria. The car damage are the reactive oxygen species, and the repairing services that try to fight them are our antioxidant mechanisms. But the biology behind this is a bit more complex. To make energy from glucose, pyruvate, and glutamine, the mitochondria of our cells employ a sequence of metabolic reactions known as the TCA cycle, which results in the generation of this deleterious reactive oxygen species that I was mentioning. But in healthy kidney cells, nothing bad happens because we count on a very efficient antioxidant mechanism that is the glutathione cycle, which uses glutamate and neutralizes this reactive oxygen species, enabling the cells to make energy and produce biomass while keeping a good metabolic balance. However, in diabetes, this balance may be disrupted. Diabetes is characterized by increased blood glucose levels, and this glucose can enter the kidney cells and hyperactivate the mitochondria that start working too much until they are damaged. In this situation, cells attempt to make ATP via glycolysis, which is less efficient in terms of energy. And when that metabolic shift happens, kidney cells may undergo what we know as glutamine anaplerosis and start investing more glutamate as an attempt to restore the mitochondrial activity, which results in reduced antioxidant capacity. Because enzymes implicated in this metabolic injury were increased by androgens, our emerging hypothesis was that androgens may disturb the metabolic balance of kidney cells. And that may be particularly important in diabetes. To confirm our proteomics observations, we then decided to validate using Western blot the observed changes in two enzymes that are implicated in glutamine and glucose metabolism. As we show in detail in the paper, we confirm the effect of androgens and male sex upregulating the expression of these enzymes involved in the breakdown of glucose and glutamine in vitro and in vivo. And our next question was, what happens in diabetes? We know from previous studies that individuals with diabetic kidney disease generally show higher renal hypertrophy and higher oxidative stress levels as a result of impaired metabolism and mitochondrial stress. We then wonder if these metabolic perturbations linked to androgens could accentuate these alterations in diabetic males. And we actually found that increased expression of these proteins was linked to increased renal hypertrophy and oxidative stress. In front of these novel findings, I was able to conclude that sex hormone-induced alterations in kidney metabolism may be the key link to understand sex differences in the biology of kidney cells, and that this link would likely play a role in the sex-specific progression of diabetic kidney disease. These observations were based on the metabolic effects of androgens over estrogens at the protein level. However, I only used male cells in this study. So then I wondered, would I have obtained similar results if I had used female cells? Or in other words, does the sex of the cells matter? As I was mentioning before, there is a growing recognition to the fact that the X and the Y chromosomes have more influence on cells than first thought. There are certain genes that escape from the inactivation of one of the X chromosomes in females, and those genes have a role on defining the female cell phenotype. Similarly, growing evidence suggests that genes from the SRY region, which is a fragment of the Y chromosome that defines the development of the male sexual characteristics, these genes can also influence the male cell phenotype in different tissues. We thus wrote a proposal to test our emerging hypothesis in kidney cells coming from both male and female donors. And I'm glad to say that the relevance of our research was positively received, and we obtained operative funding from the CHR Institute of Gender and Health. And only a year later, I got a Crescent Postdoctoral Fellowship that is supporting my work at UHN. This funding is enabling me to have some fun in the lab while studying in parallel male and female kidney cells in order to test my current hypotheses, which are the following. First, sex hormones will induce alterations in the metabolic function of kidney cells. And second, kidney cell metabolism may also be modulated by cell sex. To test this hypothesis, we study in parallel kidney cells from 6 presumably healthy disease donors, 3 males and 3 females. In the lab, we expose the cells to a media with normal amounts of glucose or with high glucose to mimic the diabetic condition. We also study the effects of sex hormones by treating them with the androgen dihydrotestosterone or with the estrogen estradiol. At the end of the treatment, we assess a set of metabolic readouts, namely glycolysis, mitochondrial function, glucose consumption, oxidative stress, and apoptosis. I will stop for a second to tell you a little bit how we evaluate glycolysis and mitochondrial function. When cells consume glucose via glycolysis, they secrete lactate to the extracellular space, which results on a decrease in the pH, or in other words, an increase in the extracellular acidification rate, or ECAR, which is our marker of glycolysis. The higher the ECAR, the more glycolysis is taking place inside the cells. In turn, to make energy in the mitochondria, the cells need to consume oxygen, and this increase in the oxygen consumption rate is what we use as a marker of mitochondrial function. In the lab, we employed this methodology named Seahorse, which enables us to monitor glycolysis and oxygen consumption in real time, after exposing the cells to a minimal media with 3 fuels of interest, namely glucose, glutamine, and pyruvate. And how do we monitor glycolysis in particular? We have our cells in a plate, and we give them glucose either in the same media or as an injection. By doing that, we achieve a baseline extracellular acidification rate as a result of lactate production. We then induce metabolic stress by injecting a drug named oligomycin, which specifically inhibits the synthesis of ATP in the mitochondria. And as a compensatory mechanism, the cell tries to make ATP by increasing glycolysis, bringing the energy to the maximal glycolytic capacity. Then we employ a specific inhibitor, which is called 2-deoxyglucose, to block glycolysis. So we can confirm that the changes that we were observing are really ascribed to glycolysis and not to other cellular metabolic reactions leading to acidification. We can also monitor oxygen consumption, or OCR. If our cells are alive, we start with a baseline degree of oxygen consumption. But what happens to OCR when we give oligomycin? It dramatically drops. If we think about it, the ATP synthase in the mitochondria employs the energy stored in the proton gradient between the 2 sides of the mitochondrial membrane in order to make ATP. This generates an influx of protons into the mitochondria that are in turn pumped out by the electron transport chain complexes while consuming oxygen. If the ATP synthase is inhibited, this gradient cannot happen, and the respiratory chain cannot work and keep consuming oxygen. So then that's why we see this decrease in oxygen. However, we can induce a continuous gradient and rescue mitochondrial respiration by injecting FCCP to the system. FCCP is a cyanide acid that attracts protons and brings them back into the mitochondria. And with this, we keep the gradient working independently from the ATP synthase, and there's a lot of oxygen consumption that brings the metabolism to the maximal respiratory capacity. That's why we recognize FCCP as a mitochondrial uncoupler, because it enables the electron transport chain to work independently from the ATP synthase. The experiment finishes with the injection of rotetone and antimycin A, which specifically inhibit complex 1 and 3 in the electron transport chain, and that helps us distinguish between specific effects of the mitochondrial respiration from other sources of respiration that we could find in the cells, such as cytosolic oxygenases. Overall, this methodology is really useful to study the metabolic phenotype of our cells, either at baseline or after metabolic stress. And after optimizing the methodology in our in vitro system, we were able to assess in real time glycolysis and mitochondrial function in male and female kidney cells. When it comes to glycolysis, we obtain these 2 different curves. The purple curve corresponds to the male cells and show their glycolytic activity over time, while the orange curve corresponds to female cells. We observe that male cells show a higher rate of glycolysis at baseline, which achieve a statistical significance after metabolic stress. Specifically, the area under the curve enables us to calculate several parameters and enable us to see in the glycolytic reserve and in the maximal glycolytic capacity in male kidney cells compared to females. By using another assay, we also observed that male cells were able to significantly uptake more glucose than female cells, suggesting that male sex in our kidney cells was associated to a more rapid utilization of glucose to glycolysis. In parallel, we were able to measure the mitochondrial function of our cells. And we observed that male kidney cells show a higher consumption of oxygen at baseline, as well as a higher rate of mitochondrial function after metabolic stress by means of significantly increased ATP-linked respiration, maximal respiratory capacity, and reserve capacity, which are other mitochondrial parameters that can be calculated by the area under the curve. These results indicate indicate that the higher utilization of glucose linked to male sex could be associated to a higher activity of the mitochondria. Our next question was if sex hormones would also alter the metabolic function of the kidney cell. To a lesser extent, we also found that sex hormones influence the metabolic phenotype of kidney cells, and probably in a sex-specific fashion. In male cells, DHT, which is represented by the red dots, but not estradiol here in green, induce an increase in glycolysis, which was significant at baseline, as compared to control-treated cells. However, when we look at oxygen consumption, both hormones were able to increase this parameter either at baseline and after metabolic stress, suggesting that estradiol could be increasing mitochondrial activity via mechanisms that are independent from glycolysis, while androgens could promote a more energetic phenotype by increasing both parameters in male kidney cells. In female cells, we observe a similar effect of androgens and estrogens on increasing oxygen consumption. However, we did not observe any significant difference in the glycolytic rate between treatments. In summary, we found that the metabolic function of kidney cells is a sexually dimorphic feature, with male cells showing increased glucose consumption, increased glycolysis, and higher mitochondrial respiration than female cells. After identifying that male cells are energetically more active than female cells, our next question was if these sex differences on the metabolic function could relate to differences in oxidative stress and cell death. We analyzed superoxide ion as a marker of oxidative stress, and we could accept our hypothesis because we observed that male cells had higher oxidative stress levels than female cells. Similarly, male sex was associated to a higher degree of early apoptosis by means of a higher higher translocation of the apoptotic marker phosphatidylserine to the cell membrane. And when it comes to sex hormone effects, DHT-treated male cells show higher levels of oxidative stress as compared to the cells exposed to vehicle or to estradiol treatment. But no differences were observed among the female cells. And a similar pattern was observed when we assessed early apoptosis. In summary, we observed that under physiological conditions, male cells are metabolically more active than female cells in terms of increased glycolytic and oxygen consumption rates, which probably results in higher baseline levels of oxidative stress and cell death. And we also observed that these changes were more pronounced in the presence of androgens. In diabetic kidney disease, oxidative stress contributes to inflammation and to the development of kidney fibrosis, which accelerates disease progression. It is reasonable to think that if male cells are more active and produce more oxidative stress, they may be more prone to injury in the setting of diabetes. Supporting this idea, we also found that under high glucose conditions, male cells show a more rapid decline in mitochondrial function compared to female cells. And because male cells clearly show a different metabolism than female cells, and this implicates the mitochondria, we wanted to further investigate how are male and female cells making energy. If we go back to our design, we can remember that we are feeding the cells with glucose, glutamine, and pyruvate. It was logical to think that if the metabolic outputs that we are measuring differ between sexes, that means that male cells are probably using these food options different than female cells. So we decided to study what is the specific contribution of each of these substrates to these fundamental metabolic sex differences. Importantly, we have seen that the mitochondria of male kidney cells like glutamine, apparently more than the female ones. On one hand, when we expose our cells to glutamine as the only food option, we see the difference in mitochondrial function being more pronounced than what we saw before. In concordance, when we remove glutamine from the media, we lose this phenotype, and male cells do not show increased mitochondrial respiration anymore. And because we still had two food options on the plate, our next question was who is responsible for this capacity of female cells to even up with the mitochondrial rate of male cells? Is it due to glucose or to pyruvate? And we had the answer in our next experiment when we saw that glucose alone still led to increased mitochondrial activity in male cells, but this parameter didn't change between sexes when pyruvate was the only food option. These interesting results suggest that relative to glucose and glutamine, female cells may be better on using pyruvate. If we go back to our cat analogy, our data suggests that while male cells may be more efficient utilizing glucose and glutamine, that may have a bigger price in terms of oxidative stress, and under certain circumstances may lead to a bigger damage of their metabolic machinery. In turn, female kidney cells could prefer to utilize other fuels and have a more quiescent and balanced metabolism. At that point, we had an idea of the inputs and outputs that distinguish how the motor of male and female kidney cells could be working, but we still didn't know at all what actually happened inside. To address this question, we collaborated with two experts in metabolomics at U of T to help us characterize the global metabolome changes that occur in our cells as a consequence of their sex. Today, I will focus on our key findings when it comes to intracellular metabolites, as they may reflect better how cells work internally. In line with our functional data, we observed that male cells had increased levels of glutamate, and also metabolites such as citrate, malate, and succinate, which belong to the TCA cycle, that metabolic process that results in the generation of oxidative species. We also examined metabolites of the glutathione cycle. To perform the antioxidant function, this sequence of reactions involve the generation of glutathione, or GSH, which is the metabolite that exits the cycle in order to neutralize the oxygen species, while oxidating itself and generating GSSG as a byproduct. Interestingly, we found that male cells had increased levels of both glutathione metabolites compared to female cells, suggesting a higher demand for antioxidant mechanisms in order to balance the increased mitochondrial activity. And one more time, we observe a certain analogy with our functional studies when we saw that reduced reactive oxygen species in female cells was linked to increased levels of pyruvate. In summary, the increased oxidative injury in male cells was linked to increased levels of glutamate and metabolites of the TCA cycle, as well as glutathione cycle metabolites. In turn, the more paused metabolism of female cells was linked to increased levels of pyruvate. While these observations are very intriguing at the mechanistic level, are they actually relevant to our patients? We never lose perspective of our ultimate goal, which is to translate our findings to the patient. Our findings suggest that in response to the diabetic condition, there is a sex-specific rewiring of metabolism that may result in a different progression of diabetic kidney disease in male and female patients. And we believe that glucose, glutamine, and pyruvate could hold the key to understand how these differences occur, and that this understanding could help us design better sex-specific treatments and maneuvers to prevent the establishment of diabetic kidney disease. And to be able to act early enough, it is crucial to capture these metabolic alterations early after the onset of diabetes and before the establishment of overt nephropathy. To test the translational potential of our findings, we leverage our collaboration with Dr. Alison Dar and the iCare research team from the University of Manitoba. iCare is a prospective observational cohort study to identify risk factors for early onset albuminuria and progression of chronic kidney disease in adolescents with type 2 diabetes. Thanks to this collaboration, we were able to interrogate metabolite levels in a cohort of First Nation male and female adolescents with type 2 diabetes, and they are age and body weight match healthy individuals. In the iCare cohort, which has been previously described, there was a higher representation of females in both diabetic and control groups. It is also worth highlighting that the median age between the two groups was remarkably similar. The median duration of diabetes was only 2.3 years. As one would expect in type 2 diabetes, the disease group shows significantly higher glycated hemoglobin and body mass index in comparison to the control group. And despite being clearly diabetic, most subjects were normotensive and display a significant but still modest increase in the excretion of albumin and in the glomerular filtration rate, indicative of an incipient degree of diabetic kidney disease. And we consider that the overall incipient disease among the diabetic patients with no other major comorbidities could make iCare cohort a unique, clean setting to study early metabolome changes influenced by sex and or diabetes. And with this goal, serum and urine samples were collected from the participants and subjected to unbiased metabolome analysis by mass spectrometry. Today, I will focus on the blood. Since we already had some in vivo data about the specific metabolites, we first interrogated the circulating levels of glucose, lactate, and glutamate. In our mice, males show higher plasma levels of the three metabolites, and this increase was more pronounced and significant under healthy conditions, especially for lactate and for glutamate. And in line with our observations from animal models, in the iCare cohort, male sex was linked to increased circulating levels of glucose, lactate, and glutamate. These consistent findings across the species suggest that overall, male tissues could be breaking down more glucose to lactate and also using glutamate differently than female tissues. Supporting this idea, and in concordance with our findings in the lab, male sex was also associated with increased circulating levels of TCA cycle metabolites, such as fumarate, as well as malate and succinate, that, if you remember, were also increased in male cells compared to females. And although this sex difference was statistically significant only among healthy individuals, the same trend could be observed among diabetic patients. We also found that the serum levels of 14 metabolites related to the glutathione cycle were increased in males. These metabolites belong to the family of gamma-glutamyl amino acids, which are a source of substrates needed by the tissues to keep the glutathione cycle working. Here we show only 4 of them as example, and as you can see, all of them display the same profile, being higher in males, correlating with each other, and also being decreased by diabetes. We speculate that this decrease in circulating gamma-glutamyl amino acids observed in diabetic patients could be due to a higher demand of antioxidant mechanisms by the tissues that are being challenged by hyperglycemia. But more studies are required to address this question. It is also worth to mention that, resembling our in vitro observations, in the human cohort, there was also a link between female sex and increased levels of pyruvate, although it was not statistically significant. In conclusion, concordant in vitro, in vivo, and human data suggest that males have a more active metabolism of glucose and glutamine. While that may enable kidney cells to produce more, to be more efficient on producing energy from these substrates, it may also predispose them to a higher damage when they are metabolically challenged, like it happens in diabetes. In turn, there seems to be a connection between pyruvate and a better metabolic balance in female kidney cells. We started this project with a problem, and we hope that the validation of our key observations in the ICARE cohort brings us a bit closer to the solution. These days, we are investigating the role of certain regulators on these metabolic sex differences, or in other words, we are trying to understand in the lab what instigates male and female kidney cells to consume glucose, glutamine, and pyruvate differently. Our goal is to target kidney metabolism in a sex-specific fashion so we can help prevent diabetic kidney disease in men and women. In terms of future directions, we acknowledge that so far we have been able to see some elements from the motors of our male and female kidney cells, but we still don't know exactly what happens inside. Our next step is to label these fuels before giving it to the cells so we can track them and identify what are their fates once they are inside male or female kidney cells. And that's why we are currently designing very exciting nutrient flux analysis with our expert collaborators on metabolomics. We are also excited to see how approaching the study of kidney sex differences from a totally different and unbiased angle is leading to similar observations. Dr. McEvoy, who completed a postdoc in our lab, has been working closely with the team of Dr. Crom, and they have generated a very unique single-cell RNA-seq dataset of human kidney biopsies from healthy and diseased male and female donors. In this figure, I'm showing what is called a varimax analysis, which is a type of principal component analysis. This analysis enables to see if 2 or more groups are distinct enough so they can be segregated based on gene transcripts that can be found inside every single cell. Reassuringly, the analysis showed that the cluster of proximal tubular cells is the most diverse between sexes, as reflected by a clear segregation between the green dots, which represent the male tubular cells, and the pink dots, which represents the female tubular cells. One more time, metabolism is the main driver of these sex differences. At the bottom of this figure, we can observe biological processes that were significantly enriched among genes that are increased in male tubular cells compared to females. And supporting our findings from other projects, most of these processes are related to the TCA cycle and the mitochondria. In front of these exciting observations, we are intrigued about their broad implications, and we wonder if similar results would emerge from the study of kidney diseases, as well as from the analysis of other tissues. If we look back at these 10 years, we realize that several layers of evidence have guided me to determine that metabolism is a crucial pillar for the study of biological sex differences. So far, I have determined that these sex differences are important in the kidney for health and in a setting of health and also in a setting of diabetic kidney disease. And due to the metabolic component, we believe that our observations may also have an impact on other kidney diseases that show a sex-based epidemiology. For example, like we observe in our cells, renal cell carcinoma and polycystic kidney disease are characterized by increased glycolysis, followed by glutamine utilization. And these diseases also disproportionately affect men more than women. Sex differences may also impact the outcomes after a kidney transplant, in which the graft can suffer from ischemic reperfusion injury after implantation and revascularization. To study metabolic sex difference that can be relevant in kidney transplant, we are developing in collaboration a model of ischemic reperfusion injury. And so far, we are seeing that in concordance with previous studies, males seem to show an increased susceptibility to injury in comparison to females. Rejection outcomes after transplant have also been to increased blood and urine levels of lactate, suggesting a higher glycolysis in the tissues. And interestingly, we are seeing that male kidney cells increase glycolysis to a bigger extent than female cells in response to hypoxia. While my research program currently targets the kidney, I hope that with your help and advice, I can learn and explore about other solid organs and investigate if they are also influenced by these fundamental metabolic sex differences. Because if we think about it, all of our tissues in the body are made by cells that contain sex chromosomes and that need to make energy in order to function, right? At the beginning of the presentation, I showed this diagram. And every day I'm more convinced that the field of sex differences has numerous applications across all solid organs. And I see how this program of research could align with the interests of many divisions of the multi-organ transplant group. I actually suspect that some of you are starting to formulate new hypotheses on how sex may influence each of these organs in health and disease. And to keep you thinking, I will finish by giving just a few examples around this idea. In the pancreas, sex differences have been detected in beta cells, with androgens being deleterious and estrogens being protective, and with male cells displaying higher oxidative stress than female cells. On the other hand, we and others have shown that male animals display increased blood glucose levels than females, and that this effect is prevented by the absence of sex hormones. This is probably important because men show a higher incidence of diabetes compared to women. Thus, our findings are relevant for diabetic kidney disease, but they could also lead to a better understanding of diabetes. And if we talk about metabolic disorders, we talk about obesity, and we think about adipose tissue. To start, there is a sex dichotomy in the distribution of fat in our bodies. While adult women have a greater percentage of total fat mass, which is mainly given by more fat accumulation under the skin, men tend to have greater amounts of visceral fat. Interestingly, studies in animal models have determined that the X chromosomes directly contribute to the increased total fat mass in females. In turn, estrogen actions in adipocytes stimulate mitochondrial biogenesis and lipid oxidation to prevent the excessive accumulation of fat in the female tissues. And some authors believe that this process may reduce their risk of metabolic disease. Indeed, they think that the decline in estrogen levels after menopause may result in increased fat content and the loss of metabolic protection, similarly to what happens in certain kidney settings. Through interactions with liver experts such as Dr. Bhatt, Dr. Sapisuchin, and Dr. McParland, I am learning that the liver is also a very interesting organ from the metabolism perspective. Indeed, the link between female sex and increased lipid oxidation has also been described in hepatocytes. And this could have implication in metabolic disorders such as fatty liver disease, or even in other liver diseases with a sex dichotomy, such hepatocellular carcinoma. Again, it seems that female tissues depend on substrates that are different from glucose and glutamine. And if we talk about energy demands, we can ignore the skeletal muscle. Increased muscle mass, energy expenditure, and exercise performance in men versus women have been associated with a higher oxidation of glucose by male skeletal muscles. In turn, female muscle cells tend to oxidize more lipids than glucose. And this, in a way, is resembling our findings for preferential food options in male and female kidney cells. And since we are talking about muscles, let's not forget about an important one. In the heart, ventricular hypertrophy has been linked to a remodeling of glucose and lipid metabolism in animal models of cardiac dysfunction. And in other studies, this remodeling has been linked to the effect of sex hormones. And these kind of studies could help us understand why men are more prone to die of ischemic heart disease and heart failure. Finally, thanks to collaborative studies with experts like Dr. Jouvet and Dr. Martino, I am also getting to know a bit more about the lung. This tissue has been under the radar for decades when it comes to sex differences, but increasing evidence supports that sex affects the incidence, morbidity, and mortality of several lung diseases, including allergic diseases, lung cancer, chronic obstructive pulmonary disease, and pulmonary fibrosis and hypertension. It is actually believed that estrogen plays an important role in lung cancer. Among the 3 types of estrogen receptors, estrogen receptor beta is the most expressed in several cell types of the lung. And this expression is even higher in cancer lung tissues compared to healthy tissues. And to the point that the signaling through estrogen receptor beta could become maladaptive for the cells. The potential link between metabolism and the sex dichotomy in lung diseases is still a mystery. And even more mysterious are the potential effects of the sex of the cell in each of these organs, which remains largely unexplored. Hopefully you will agree with me that to investigate these mysteries and solve the puzzle of sex differences, we need to team up and have a crosstalk, the same way that our organs talk to each other. And to achieve that, I truly believe that there is no better place for a scientific program on sex differences than a prominent and multidisciplinary institution as UHN. I also want to emphasize the importance of nationwide training programs like Crescent, which is doing a great job enhancing the kidney research capacity in Canada by supporting young researchers, by fostering interactions between clinical and basic scientists, and also by facilitating networking and collaboration. And since we are talking about networking, we all remember how much fun it was attending to conferences. And I confess I'm totally biased, but I personally recognize the value of this annual meeting of the Organization for the Study of Sex Differences, in which other fanatics of the field are reassuring me that a better understanding of the physiological sex differences will make us better on understanding and treating diseases. Hoping that today's talk has been useful for communicating this idea, I will finish by thanking you all for your attention, with a special mention to the Crescent and the CIHR programs, which are enabling me to pursue my scientific goals. Now I'm happy to answer any questions.
Thank you, Sergi. That was great. Can you turn on your camera, if you don't mind, and I'll ask the participants if they would have any questions. I don't see any hands up, but you could unmute yourself and ask. Oh, Marcelo Cipel has a question. Marcelo, please go ahead.
Hi, can you hear me okay?
Yes.
Yes.
Yeah, that was really a fantastic and very comprehensive talk. I was just wondering if you had looked at, I haven't, you talked a lot about a lot of metabolites there, and I haven't heard you talking about itaconate, which seemed to be a relatively hot metabolite recently, but it's very key in terms of modulating the inflammatory response of mitochondrial injury. So I just wondering whether you looked at that and the differences between that metabolite in response to injury between male and females. And in addition to that, have you looked at inflammatory downstream markers of that? Because I think at the end of the day, that's what's causing the injury as well. And so, IL-1 beta and IL-8 and NLRP as downstream effects of mitochondrial injury in the differences between the two.
So in answer to your question, no, we haven't looked at that, but after this comment, we probably will. In the cell metabolome, the detection of metabolites depends on certain like settings where we try to favor the detection of metabolites belonging to different processes due to their structures. So when we perform our metabolome analysis, we were very focused on certain metabolites, on favoring the detection of several metabolites of the TCA cycle and the glutathione cycle. And when we interrogated the human metabolome, we also share our interest for a specific lists of metabolites. But it's true that we want to move towards more disease models in our in vitro systems, and these cytokines that you have mentioned are linked to high glucose injury. That is one of the things we are working on, and are also linked to certain metabolic regulations such as KDM6A, which is a target that we are exploring. So definitely connecting metabolism to inflammation is one of our goals, and I definitely appreciate this suggestion because it could be a really interesting lead, because as you said, ultimately we need to link it to injury pathways. Yeah.
Okay, next Anand, then Atul, then Myron.
Hi, thanks for your talk. I thought that was really interesting. So I guess now there's a lot of information there, so forgive me if I didn't understand it all, but I mean, my impression is that there are both extrinsic factors and intrinsic factors that account for the differences. And I guess my question is could you speculate or comment on, in a transplant situation, what do you think is the biology that's going on when we transplant a male kidney into a female recipient, or a female kidney into a male recipient? And does that have any importance on the natural history of the allograft performance post-transplant, and particularly in the setting of the recipient's disease, like diabetes, which is common, or if it's a kidney that comes from a donor with high-risk features, you know, and are there ways that you could use some of the resources we have here in terms of biobank tissues or small animal transplantation models to answer some of those questions?
Yes, absolutely. Like, obviously, it's hard to speculate about a specific hypothesis that could be tested in this setting, but it's clear that I'm going to try to do my speculation based on metabolism, which is the topic that is emerging in all the studies we are doing when it comes to sex differences. And we would be talking about transplanting a kidney from a male, for example, that has XY chromosomes into a female that has XX chromosome in the rest of the body, and presumably higher estrogens than the male. So obviously, I would assume that the kidney coming from a male, regardless of the setting, would have a higher propensity to develop certain metabolic stress, because we are seeing that these differences in metabolism are likely more ascribed to sex chromosomes than sex hormones. So as long as the kidney is XY, my hypothesis would be that this mitochondrial injury followed by glycolysis that likely happens after certain transplants that have been suffering from the lack of oxygen. I would think that an XY male kidney would be more prone to this injury regardless of the recipient. And for that reason, it would be really interesting to perform studies in animal models and also access samples from the biobank to test this kind of hypothesis, because we have a lot of targets representative of these metabolic changes.
And then Sergi has also cited a study which shows clearly that female recipients tend to do better in the long term and retain their grafts longer, and that's conserved in animal models and tolerate ischemia reperfusion better compared to males. So yeah, the biobank would very nicely lend itself to further studies that Sergi is interested in this regard. Then I'll ask Atul to go next, please.
Thanks. Thank you, Sergi. That was a really nice body of work that you presented. Congratulations. So my question is, you know, these patients with diabetes, they're on a lot of drugs often that affect metabolism, like metformin or these SGLT2 inhibitors or other drugs. And is there a sex difference when in terms of the response to these drugs?
We have not directly examined that in the lab, and one would, and there's a lot of studies out there showing sex difference in the expression and distribution of not only SGLT2 but several glucose transporters and also glutamine transporters in kidney cells. But I'm not aware that the effects of the sex differences on the effects of this drug has been examined directly in a sex-specific fashion. And even though that was the case, our studies seem to suggest that what is important for a better management of the injury at the kidney cell level is to control the metabolic homeostasis from what happens inside the cells. So in other words, how to use glucose better rather than how to prevent glucose to enter the cell, because it's still a necessary substrate. Along these lines, we are in the process to obtain more comprehensive clinical data from our collaborators and hopefully this information about drugs can help us answer some of these questions that arise from your comment in the near future.
Thank you.
No problem.
Myron.
I also congratulate you for a very clear talk in an area that is generally difficult for most of us to follow. My questions relate to different cell types found in the kidney. You implied in your talk that the proximal tubule cells had the greatest differences, yet the kidney is full of many other cells, including endothelial cells, myeloid cells, etc. What makes the epithelial cells more prone to these differences? And when you study plasma levels from patients, how do you know which cell type is influencing the differences in the plasma levels that you studied by metabolomics?
Yes. Okay. So in answer to your first question, which refers to the preference for kidney tubular epithelial cells, this is, of course, like we are biased here, right? And we are biased by the disease of interest that was our focus when the project started, which is diabetic kidney disease. So these studies in sex differences are pointing us towards metabolism, and because it is known that the tubular cells have a lot of mitochondria and are constantly importing and exporting glucose and other substrates for reabsorption and transport functions that are key for the kidney physiology. It's a very metabolism-rich setting in which there's probably a higher likelihood to identify sex differences, not only under physiological conditions, but when we challenge the cells with something like high glucose that is directly linked to metabolism. But that doesn't mean that this is not important or potentially applicable to other cell types. For example, something I would love to do is to apply the SILAC proteoma study that we did in the past to other cell types of the kidney that also express sex hormone receptors and have X and Y chromosomes, obviously, right? It was just a matter of priorities, but I do believe that there's also applications. For example, the podocyte also is from epithelial lineage, right? So there could be some resemblances there. And in response to your second question, we actually cannot say that there's a link between our blood data and the kidney. I haven't shown it today, but we have some metabolome data from the urine. So obviously by doing some correlations and connections between the blood data and the human data, one could infer that what is in between could be the kidney, and some changes could be ascribed to the kidney activity, and the urine would be the output. But still to be able to pinpoint the kidney is doing that in a male or a female, and that results in this metabolite change in the blood or the urine. We would need all these kind of experiments that I was saying about tracing substrates through mass spectrometry-based methods. And these type of experiments can be done in vitro, but also in vivo. So I think this kind of evidence is needed, and it's what I would love to focus my studies in the near future. And since we cannot really do this kind of heavy isotope leveling of food that the humans eat, and we cannot conduct this experiment in human, it would be nice to have somehow prospective studies that enable us to correlate findings in biopsies from living healthy and diseased donors with circulating data.
Thank you. I have another quick question with regards to signaling within cells. I know certainly when macrophages are exposed to pro-inflammatory signaling, they switch from aerobic to anaerobic metabolism, glycolysis mostly, which is also known as the Warburg effect. Does the same thing happen in proximal tubular epithelial cells, and what impact would sex have on that switch?
Yeah, that's a very good point. Actually, we are scared to call it Warburg effect because it seems that there's not enough, like, let's say, reviews or like high impact papers, like, but there's a few of them that suggest the name of Warburg effect. But I truly believe there is. I truly believe the answer is yes. I think that it's the same concept, that even in the presence of oxygen, there are certain circumstances that make the renal tubular cells make more glycolysis that they should be doing. And actually, we started getting interested on that when we had our proteomics study, and then we held some journal clubs, and we presented actually papers on activation of non-macrophages, but I think it was T cells, and the activation was linked to a higher glycolytic, to a switch towards a higher glycolytic activity. And then it's when we started to really explore these glycolysis-related processes. So I think we can talk about it, that because I am not showing all the readouts today, but overall, it seems that the male cells are kind of, if you want to call it, more aggressive or more proliferative. And we have seen in cancer, right, that cancer cells, when they undergo Warburg effect, they also are kind of classified as more aggressive. So I think that this more aggressive phenotype and higher glycolysis apply to more settings rather than cancer. It's just that they have not been explored enough, so people actively call it Warburg effect, but I think that will happen in the future. Yeah.
Okay, great. Thank you very much. And you see lots of interest. Sergi, Stephen has a couple more questions, but we don't have time and we have to end now. But Stephen, we'll get back to you on these questions. Thank you very much, everyone, for attending and participating and asking questions. And let's thank Sergi for an excellent presentation.
Thank you.
Have a good day.